All right, welcome to the next installment of my short series on metabolism. If you haven't watched the introduction of metabolism, I think it's worth your time to do so. For us, I want to go back and visit this idea that metabolism is a story of carbon oxidation. And if we understand that, then we'll understand where the energy is in the macromolecules that we eat, the carbs, the fats, the protein. And I want to focus today's discussion on the central hub of all of these things, which is acetyl-CoA. There's acetyl-CoA. We see the acetyl unit in white, the coenzyme A in blue. This little molecule is really fascinating because of what it can do in terms of potential energy. I don't want to detail the steps of Krebs cycle or beta oxidation in this video. That will be a different video. But I do want to just briefly highlight the fact that the acetyl-CoA that's generated in our bodies is going to join with a 4-carbon molecule called oxaloacetate. So that's going up. These two are going to join together, and we end up, of course, the coenzyme A leaves. We end up with a 6-carbon molecule. I get that it doesn't look like that. I know it's not linear, but we're just counting carbons right now. A 6-carbon molecule called citrate. This is the citric acid cycle. That citrate is going to go through a series of redox steps in which 2 of the carbons are going to be completely oxidized to form CO2, right? That's going to be 2 CO2, and this represents carbon oxidation. And of course, in the meantime, I'm stripping electrons off of carbon. And so what do I get out of this process? I'm going to get 2 NADH molecules. And I introduced NADH to you in the previous video. We're also going to generate an ATP. And again, I'm not looking to go into detail in this video on this process. But we get an ATP here. Over here on the back end, we get an FADH2, which is very similar to NADH, only it doesn't carry as high energy electron. And then one more on the back end, one more NADH. And so we see that as we go around the Krebs cycle, we end up with 3 NADH, 1 FADH2, and 1 ATP. Why do I bring this up now? Because I want you to recognize the energy in that little acetyl-CoA molecule. In the electron transport chain, we're going to see that every one of these NADHs is going to has the potential energy to produce about 3 ATP. So that's going to translate 3 times 3, that's 9 ATP. The FADH2, one of those will make about 2, so we've got 2 ATP here. And then, of course, the 1 ATP that we generate in the cycle, that's a total of 12 ATP for just one acetyl-CoA. So as we see that as a central role in metabolism, I want you to recognize, how do we get to acetyl-CoA? Well, from carbs, we get there through 2 steps. Glycolysis, and then we pass into the mitochondria, which is a double membrane organelle, so we'll draw 2 lines there. And then the step that we talked about previously, sometimes known as the bridge step, this is pyruvate oxidation. That gets us to acetyl-CoA. What about fats? Fats are broken down through a process called beta oxidation to generate acetyl-CoA. We could also add protein, but here I have to be honest. It depends on the amino acid. The different amino acids will plug into this whole cycle of metabolism in different places. But certainly there are some that will end up here at acetyl-CoA. So this molecule itself represents a key hub of all of the redox reactions. And with that one molecule, there's a lot of potential energy in there. Now, we're gonna go through and we're gonna talk about each one of these steps individually. Glycolysis, pyruvate oxidation versus lactic acid fermentation. We're going to talk about fats and beta oxidation, and then we're going to show you how all of these electrons are run through the electron transport chain to create the ATP that we mentioned here. This is more of a holistic big picture, but I would like to recognize too one of the key aspects of the keto diet. The keto diet has gained a lot of popularity in recent years. It's a really good way to lose fat fairly quickly because by not eating carbs, it forces your body into a chronic fat burning state. Um, don't want to talk too much about the diet itself, but rather what it's named for. And that is the state of ketosis that the body enters when you're not eating carbs. And I'd like to talk just a little bit about ketosis and why it's worth mentioning. Ketosis has everything to do with this molecule, this acetyl-CoA. And for reasons that I'm not going to go into in depth here, because your body is primarily burning fats through beta oxidation, you end up with a lot of acetyl-CoA. And the Krebs cycle gets a little bit clunky because it's worried about running gluconeogenesis, some other things, again, things that we don't necessarily want to talk about here. But what happens if we have an increased buildup, and I'm talking about significant increased buildup of acetyl-CoA? We'll just, this molecule H3CC double bond O dash coenzyme A. Well, when I get high concentrations of acetyl-CoA, acetyl-CoA starts to stick to other acetyl-CoA in ways that it normally wouldn't. And so if I add to this another acetyl-CoA, these two molecules will actually stick together. They'll actually combine. I'm not going through all the detail on this, but we end up with this molecule here, beta-hydroxybutyrate. Beta-hydroxybutyrate is one of the principal ketone bodies produced by this process. And there's a couple of things that I want to point out. This functional unit here is a carboxyl group, which means at physiological pH, this H is going to leave as an acid, and this is going to carry a negative charge. And so in a state of diabetes, or you can even see this with chronic alcoholism or just starvation, you can actually induce a state of ketoacidosis because the production of these ketone bodies increases the concentration of acid inside your blood. For most of us, though, certainly a keto diet, if there are no other underlying health concerns, it's really difficult to produce enough ketone bodies to enter a state of ketoacidosis. But we can enter a state of ketosis. And a state of ketosis is just the production of these ketone bodies, but in lesser amounts. There's another ketone body that I think is really interesting, and this can be broken down further into acetone. Yeah, fingernail acid or fingernail polish remover. This is one of these intermediate ketone bodies that's going to be produced in this process. That's really interesting because the body doesn't necessarily like acetone and works to get rid of it. We can actually lose acetone through our lungs. This will exit the body as we breathe. And oftentimes people in ketosis, certainly people in ketoacidosis, you can smell on their breath. Some people describe it as rotten apple smell, something like that. This is just the acetone actually leaving the body through the lungs as it becomes volatile and tries to exit the body. Of course, ketone bodies can also be eliminated through the urine. This is a common test for diabetics, or certainly anybody that's on a keto diet. They want to be able to track the concentrations of ketone bodies, and this is a simple urinalysis. But in that sense, that's the beauty of how the keto diet works. Remember, at the beginning, we said that every single one of these acetyl-CoAs was worth 12 ATP. But on a keto diet, I'm taking these acetyl-CoAs where there's a lot of potential energy into them, and I'm converting them into structures like acetone, like beta-hydroxybutyrate, simply because these things are binding together in ways that they normally wouldn't. And in this sense, I'm able to get rid of these high energy molecules simply by breathing, simply by going to the bathroom. These calories are ending up in my breath and in the toilet. Anyway, I just wanted to provide that as a landscape from which we can now start to explore. We will detail glycolysis, we will detail beta oxidation, we will detail the electron transport chain and how we take these electrons and actually turn them into ATP.